Anatomy and Physiology 1, Chapter 2, The Chemical Level of Organization. Now, thank goodness this is not a chemistry class, so we don't have to go crazy super deep, but we do need to have a basic understanding of some chemistry and biochemistry principles in order to really go any further with physiology. So we'll start first with what chemistry is all about. So this is the science that deals with the structure of matter, which is anything that takes up space and has mass. And the study of chemistry is also going to include the structure of atoms, which are the smallest, most stable units of matter, basic chemical building blocks, and how atoms can combine to form more complex structures. So again, reiterating what matter is, it's anything that takes up space and has mass. It's made up of atoms, and atoms can join together to form chemicals that have different characteristics. Again, chemicals are what the body is made of, so we do need to understand a little bit about this chemistry to really get into the physiology. Chemical characteristics can determine physiology at the molecular and cellular levels, and we'll be talking about the cell in the next chapter, chapter 3. So atoms are made of subatomic particles, protons, which have a positive charge, neutrons, which are neutral, and electrons, which are negative. So when we look at a drawing in a book of an atom, you'll find that protons and neutrons are at the center of the atom, also known as the nucleus or hub, middle of the atom, and the electrons will be found orbiting around the outside of that central nucleus. Atomic number is the number of protons an atom has. The nucleus, as we just mentioned, is going to contain the protons and neutrons together at the core. In the electron cloud, in some books we're going to see an electron cloud, and I'll show you a difference between that and the electron shell. But the electron cloud is the spherical area that's going to contain the electrons. And the electron shell is a two-dimensional representation of the electron cloud. And most of the figures we're going to use are going to have the electron shell. So here you can see the electron cloud on the right, which kind of looks like a hazy cloud representing where the electrons are found. And then on the left we have the electron shell, which is a two-dimensional representation. So we can see the actual atom, or excuse me, the actual electrons here and here in that shell. And then notice at the middle, which we call the nucleus, we can see the pink colored protons and the gray colored neutrons. Remember again that protons are positively charged and neutrons are neutral. So an atom is made up of protons and neutrons, again, located in a central nucleus. The nucleus is surrounded by the electrons. Now the electrons hang out with this nucleus because protons are positive, and positive plus neutral or nothing equals an overall positive charge in the nucleus. The electrons are negative, and what happens when negative and positive are near each other? They attract. So the electrons are going to be attracted to the nucleus, which will keep them around orbiting. An element is a pure substance made up of atoms of the same kind. And as we mentioned previously, the atomic number, or number of protons in an atom, is going to help determine its chemical properties. There are 92 elements that exist in nature. Atoms of each element can be distinguished by their specific number of protons. So again, each atom has a specific proton number, and that's part of that atom's identity. So it's not going to change. Carbon, for example, has 6 protons, and we can see 6 protons here in our chart for carbon. That's never going to change. That's what makes carbon carbon. But the number of neutrons can vary in the same atom, and we call those variant forms of an element isotopes. So an isotope is a variant form of an atom that differs only by neutron number. Because remember, we can't change the proton number. That's specific for that atom. So if we look at the chart down here, we can see that there are 3 forms of carbon, or isotopes, in this chart: carbon-12, carbon-13, and carbon-14. We know carbon has 6 protons, and you will see in these isotopes 6, 6, and 6. That's not going to change. If we know the number of protons in the atom, we also know the number of electrons, because the proton number and electron number will match in the atom. So since we know there are 6 protons in carbon, there will also be 6 electrons in carbon. And if we look across at these isotopes, we see 6 electrons, 6 electrons, and 6 electrons for all 3 types of carbon. Notice where the difference is in the neutrons, which again is the definition of isotope. Same or variant forms of an element differ only by neutron number. So carbon-12 has 6 neutrons, carbon-13 has 7, and carbon-14 has 8. So these guys have identical chemical properties and are alike, except for that neutron number, which is going to change their mass. So they're going to be different in mass. Okay, so next we talk a little more about electrons. So electrons in the electron cloud can determine the reactivity of the atom. The electron cloud contains what are called shells or energy levels, and each of those energy levels can hold a limited amount of electrons. Again, thank goodness this isn't chemistry. If you take chemistry, you'll need to know about all those energy levels and how many electrons go in each one, which is super, super fun. It's not fun at all, not at all, not even a little. But we do need to know, for purposes of anatomy, we need to understand about bonding. And in order to understand about bonding, we do care about the outermost shell of the atom. The outermost shell in the atom is called the valence shell, and it is going to determine bonding. So an atom with an unfilled valence shell is more likely to react with other atoms to try to obtain a full outer shell. Atoms with full outer shells already are stable and do not readily react with others. So in most, not all, but in most atoms, the outer shell can hold 8 electrons. So if an atom lacks any of those 8 electrons in its outermost shell, the only one we care about right now, it is going to be more likely to form bonds with other atoms. And in those bonds, they will either share, gain an electron from another atom, or lose an electron to another atom. So let's go into bonding now. The chemical bonds we're going to look at involve, again, sharing, gaining, or losing electrons, and there are 3 major types of chemical bonds. Whenever we have bonds, this is going to help us to form molecules and also compounds. So the 3 major types of bonds we're going to go over are first ionic, second covalent, and third hydrogen. So let's start with ionic. Ionic bonds are a bond between atoms of opposite charges. So let's go through that line by line. So first of all, we need to know what an ion is. An ion is an atom that has a charge. So either a plus or a minus beside it. In a bond, like an ionic bond, one atom is known as the electron donor. So it's going to give one or more electrons away. It's going to donate them. And when it gives away those electrons, remember electrons are negative, so if it gives away negativity, it's going to become more positive than it used to be. So that means it will become a cation. A cation is a positively charged atom. So remember, when we give away some negativity, give away an electron, that's going to make us more positive than we used to be, so we're going to have now a positive charge. So that atom is a cation. And one little tip to help you remember cation, the T in cation reminds us of a plus sign. So that T should make you think of a plus or positive charge on that atom / ion. Another atom in the ionic bond is going to be what's called the electron acceptor. This guy is going to take the electrons. So when we take electrons, we're taking on extra negativity. So that's going to make the atom more negative than it used to be. So that atom / ion will be called an anion. An anion is a negatively charged atom. So a way you can remember that is there's an N in anion, and N makes us think of the word negative. So N negative, this atom is going to have a negative charge. Ionic bonds, again, are defined as attractions between the cation, which is the positive ion, and an anion, which is our negative ion. So let's take a look at a picture or figure from a book on this. So this is how an ionic bond forms. So in this picture, we can see at the top a sodium atom, and we can also see at bottom a chlorine atom. Now let's look at the outer shell of the sodium. Remember, that's the valence shell that likes to have 8 in it. And you can see, represented by the yellow circles are the electrons we have right now. There is only one electron in sodium's outer shell. So it's a ways from being full. If we have an outer shell that has 8 in it, then the atom becomes stable. So let's look down at the second shell of sodium, second down. And in this one, there are 8 electrons. In chlorine, the outer shell has 7 electrons, and this open circle represents where we don't have an electron, but we could have one, and if we did, that would mean that our outer shell would be full and we would be stable. So chlorine only needs one electron to become stable. So these guys are actually a perfect pair. If sodium gives up or donates this electron to chlorine, who will accept it, then that means that the second shell down will become sodium's outer shell, and it will be full, which will make sodium more stable. So that would look like this. So in this picture, sodium has donated that extra electron to chlorine, and now the second shell down is its new outer shell, and it has 8 in it, making it more stable. Sodium is happy. Since sodium gave away that extra electron, gave away some of that negativity, sodium became more positive. So now we have a positive sign next to sodium, which means it's a cation now. Chlorine accepted that extra electron from sodium, as we can see it did right here in this picture. And when chlorine accepted that extra electron, accepted that negativity, chlorine became more negative. Now we call it chloride. So now we have a positive sodium cation and we have a negative chloride anion. What happens when positive and negative get near each other? They attract. So sodium chloride will attract to form an ionic bond. Again, an ionic bond is a bond between two oppositely charged atoms. We now call this the compound sodium chloride. Okay, so next are the covalent bonds, and covalent bonds are the strongest of the three. So we would rank covalent as 1 as far as strength. We would then rank ionic in the middle, and we'd rank our last one, hydrogen, as the weakest. So we're going to get to that in just a minute. So covalent bonds are strong bonds that involve shared electrons. So remember, in ionic bonding, we didn't share electrons. We either donated or we accepted. There was no sharing. So in covalent, we're going to share. One electron is donated by each atom to make the pair that is going to be shared. Sharing one pair of electrons is called a single covalent bond. Now, if you go on and take chemistry, or you have taken chemistry, you know that there are double and triple covalent bonds, which we're not going to go there, but single covalent bond is good enough for now. So in the covalent bond world, we can have nonpolar covalent bond or a polar covalent bond. Nonpolar covalent bonds are bonds that have an equal sharing of electrons between the atoms. These atoms are going to have an equal pull on the electrons, so we're going to share equally and fairly. We call that nonpolar. So this happens, for instance, between two atoms of the same type. So, for example, two hydrogen atoms may share a pair of electrons equally. The electrons are going to spend the same time with one hydrogen as they spend with the other. So that's going to mean equal or fair sharing of electrons. Nonpolar covalent bond. Polar covalent bond is an unequal sharing of electrons because one atom has a stronger pull on the electrons than the other. So we call this a polar molecule that forms in this type of bonding. And an example of this would be water, and I'll show you a picture of that in just a second. So polar covalent bonds is unequal or unfair sharing. In other words, somebody's going to hog the electrons. And in water molecules, we know water is H2O. Oxygen is big and strong, and it's going to have a stronger pull on the electrons than the hydrogens will. So oxygen is going to hog the electrons, making it unequal sharing. So here's a picture of a water molecule. Kind of looks a little bit like a Mickey Mouse head. We've got a big oxygen atom. And then we've got, and when I say big, we're talking about atoms, so you guys know big is not big. Because the oxygen is stronger, or has a stronger pull, it's going to spend more time with the electrons than the hydrogens will get to spend with the electrons. Because the oxygen will spend more time with the pair of electrons, remember electrons are negative, this is going to make oxygen slightly negative. And we can represent that by this little symbol that kind of looks like a broken 8. And that means slight negative charge. So because it's spending time with two electrons, it's going to have a slight negative charge because those electrons are hanging around that oxygen so much. The hydrogen atoms are not spending as much time with the electron. So because they're not spending that much time with the negative charge, they're going to be slightly positive, as you can see here and here. So this makes water a polar molecule. positive on one side and negative on the other side. So when we put a bunch of water molecules together, they're going to form our third type of bond called a hydrogen bond. Okay, so in the hydrogen bond, these are weak polar bonds between adjacent molecules based on their electrical attractions. These are going to involve attractions between a slight positive charge and a slight negative charge. So we see that in water molecules. So in this picture, there are multiple of our little Mickey Mouse heads or water molecules. And remember how we just talked about that oxygen is slightly negative, as symbolized here, and hydrogen is slightly positive, as symbolized here. So the slightly negative oxygens are going to be attracted to the slightly positive hydrogens. You can see the same thing here, slightly positive hydrogen attracted to slightly negative oxygen. And those slight weak attractions between the hydrogens and the oxygen is going to cause what we call hydrogen bonding. Hydrogen bonding in water can cause what's called surface tension, and surface tension acts as a barrier on the surface of water. This can allow insects to walk on the surface of water or water to bead up on plants' leaves or even on your skin, like when you get out of the shower. Okay, so moving on to chemical reactions. In a chemical reaction, either new bonds are formed or existing bonds are broken down. In a reaction, we're going to have what are called reactants. These are, I like to think of them as the ingredients. These are the materials we put in, or the ingredients we put in to make something. And then we've got the products, which are the materials coming out of a reaction. So materials going in are reactants, and products are the materials coming out of the reaction. Metabolism is defined as all the reactions that are occurring at one time. Energy is defined as the capacity to do work. Work is moving an object or changing its physical structure. Kinetic energy is the energy of motion, so walking, running, swimming, flying, all kinetic energy, energy of movement. And potential energy is stored energy, and it can be stored based on position. So if you imagine a ball at the top of the hill, it has not actually started rolling down the hill yet, but it could. So there's potential for it to move, but it hasn't. So that ball would be storing potential energy. If the ball begins to roll, then we call that kinetic energy. So back to reactions. The types of chemical reactions we're going to look at are decomposition and synthesis. Decomposition reactions, or catabolism, is when we break chemical bonds. For example, this AB would be broken up into A and B separately. AB broken up into its components, A and B. So when we break bonds, and we can use a hydrolysis reaction to do that. In other words, add water. And we see this happen when we put sugar in water and stir it up. That water can break up that sugar and help it to dissolve, and then the water tastes sweet. Synthesis reaction is when we make something. We form chemical bonds, and we call that anabolism. So decomposition, breaking things down. Anabolism, building things up, making new things. So this would be like A plus B equals AB. A plus B, two individuals, will combine to make AB. And we can do this using dehydration synthesis, where we take water out and we're able to make something new. So we're going to go back to hydrolysis and dehydration synthesis in a little while and go over it again. So decomposition, breaking things down, catabolism, synthesis, anabolism, building things up, making new things. Okay, so in biochemical reactions in cells, they do not occur on their own. They do not occur spontaneously. Activation energy is how much energy we need to get a reaction going. So how much do we have to put in to get the reaction started? Enzymes are protein catalysts that are going to make it easier and quicker to get the reaction going. So enzymes are wonderful. They're going to speed up reactions by lowering the amount of activation energy we need to get the reaction going. For example, we can see in this figure, in this chart, we have energy going up and the progress of the reaction going to the right. So in any reaction, as we talked about before, we need reactants, which are the items going in. And again, I like to think of these as the ingredients. This sharp hill represents how much energy or activation energy we need to put in to get the reaction going, which looks like a lot, by the way. And then we end up with a stable product or the finished product. Okay? So if we use an enzyme, we can actually lower that hill and make it easier, which is represented by the red hill here. This is how much energy it would take to get to the product using an enzyme. Now I know it's slightly lame, but I have an everyday example I like to think of when looking at this particular concept. So if you wanted to make a cake from scratch, you would need a lot of ingredients, and you'd need to put in a lot more time and focus to get to that stable cake product. So the reactants in the homemade from scratch cake are going to include a lot of ingredients: oil, eggs, flour, sugar, baking powder, etc. Lots of ingredients. And then this hill represents the amount of time, the amount of ingredients, the amount of concentration and measuring to do it just right to end up with a beautiful, from scratch, stable product cake. And that's great. But the human body always wants to conserve energy, so we're going to try to do things easier if possible. So in our everyday example, we could make a cake from a cake mix. We're going to end up with a similar stable product, but it's going to be a lot easier. So our cake mix represents using an enzyme. Okay, so the reactants or ingredients are less. The box of cake mix and a few little things we add, and it's kind of hard to mess it up. So this hill represents the amount of energy to get to our cake if we use a cake mix. So less energy, less focus, less ingredients, and we will arrive at a similar product. Nutrients are essential molecules we get from our food, and metabolites are molecules that we either make or break down in our body. These are a byproduct of our metabolism, which a metabolism, remember, is all the chemical reactions going on in the body. Organic compounds include molecules that contain carbon and hydrogen as their primary components. And there are 4 big organic compounds of life: carbohydrates, proteins, lipids, and nucleic acids. We're going to come back to those and go into them in a little bit more detail. Before we do, we're going to talk a little bit about water. Water makes up 2/3 of your total body weight. And hydrophilic and hydrophobic compounds are defined as either hydrophilic Hydro meaning water, philos meaning loving, so hydrophilic, water loving. These are things that interact with water, so things that mix with water well. Glucose, sugar, mixes with water well. Hydrophobic is water fearing, so these are things that do not interact well with water, like oil, for example. Whenever you put oil with water, the two separate. pH is a measure of the concentration of hydrogen ions in a solution. Neutral is 7 on the pH scale. Acidic is 0 to 6. And basic would be 8 to 14. The pH scale has an inverse relationship with hydrogen ion concentration. More hydrogen ions means lower pH. Fewer hydrogen ions means higher pH. So an acidic pH is lower than 7, as we mentioned before, 0 to 6. And it is going to have a high hydrogen ion concentration and a very low hydroxide concentration. Basic or alkaline pH is higher than 7, so 8 to 14. It's going to have a low hydrogen ion concentration and a high hydroxide concentration. The pH of human blood ranges from 7.35 to 7.45. That would be considered the homeostatic range, like we talked about homeostatic ranges back in chapter 1. So here's the pH scale, and you can see from the left leading up to neutral, these are the acids. And some examples of acid would be hydrochloric acid, that's way down there. Beer, vinegar, wine, pickles, grapes, tomatoes, things that are a little tart or a little sour are typically going to be found on the acidic side. As we move to the middle at neutral, 7, we see pure water and blood. And as we go from 8 to 14, these are the bases, and will include things like ocean water, bleach, ammonia, oven cleaner, all household things that are basic. So an acid gives off hydrogen ions. We call that a proton donor. So don't confuse this type of proton with the first type we talked about in the atom, that the positively charged proton. Proton meaning hydrogen ion here. So acids are hydrogen ion donors. They add hydrogen to a solution. Strong acids are going to dissociate or give off hydrogens completely, give off a lot of hydrogen. Bases are hydrogen ion acceptors, and they will remove hydrogen ions from a solution. Strong bases dissociate completely in solution. Weak acids and weak bases do not dissociate completely, and they will help to balance the pH. Cells are kept close to pH of 7 by buffers, and buffers are things that will resist pH change. They will accept hydrogens when there are too many, and they will donate them when there are not enough. And this is what's going to help to keep our pH within homeostatic range. Buffers are not perfect. They're not foolproof. Transitioning to organic molecules. These are going to contain hydrogen, carbon, and usually oxygen. They have covalent bonds and are going to include the 4 carbohydrates, lipids, proteins, and nucleic acids. So let's start first with carbs. Carbohydrates are going to contain carbon, hydrogen, and oxygen. These are delicious, you know, bread, cake, cookies, refined sugar, I'm picking all the unhealthy ones because, you know, they taste so good. But there are also other healthier ones like grains, like rice and whole wheat. Monosaccharides are simple sugars. These are going to include glucose and fructose. So mono meaning one. Disaccharides are two monosaccharides combined by dehydration synthesis, which joins the monosaccharides together. Some examples of disaccharides include sucrose, which is just table sugar, what we put in our coffee or tea, and maltose. Polysaccharides are made of many sugars condensed together or connected by dehydration synthesis. And this is going to include glycogen, starch, and cellulose. So here's a picture of dehydration synthesis. I promised we would come back to it. Dehydration synthesis is also called a condensation reaction, and this is how we form more complex sugars. So in this picture, we have on the left a glucose, and on the right, we have a fructose. So these are two monosaccharides. And if we want to combine these two monosaccharides to make a disaccharide, then we will need to dehydrate them or remove water. And if you look between the two, you can see the components of water, OH and H. If we combine those, that's H2O. So if we remove that water or dehydrate it, this will link the two together to create a disaccharide or double sugar called sucrose, which is table sugar. It's kind of cute. It looks like they're holding hands. Okay, and then in hydrolysis, hydrolysis is when we add water to break things apart. So here we have sucrose or table sugar, and if we want to break the sucrose apart back into its two individual monosaccharides, then we can just add water. So if we take the sucrose and add water, which we call hydrolysis, that will break the two back apart into the individual monosaccharides, glucose and fructose. Lipids. Lipids are mainly hydrophobic molecules such as fats, oils, and waxes. They're made mostly of carbon and hydrogen atoms and are going to include fatty acids, eicosanoids, glycerides, steroids, phospholipids, and glycolipids. Phospholipids we're going to hear about again because they are so integral in the cell's membrane. If you recall from your biology, phospholipids look like so. They're made up of a hydrophilic, which means water-loving head, and hydrophobic tails. Hydrophobic meaning water-fearing. And so when these guys group up side to side to side, they can start to form a membrane. But remember, cell membranes are a bilayer, a double layer of these hydrophilic heads. So we're going to revisit that phospholipid bilayer again in Chapter 3 just to remind ourselves what the plasma membrane looks like. Proteins are the most abundant and important organic molecules. They contain basic elements, and they're made up of individual amino acids joined by peptide bonds to form proteins. So proteins are long chains of amino acids, again, joined by peptide bonds, and there are four main protein shapes. So proteins can come in different shapes, and these shapes are really important to the function of the proteins. So primary shape is the simplest. Primary proteins are a linear, straight chain of amino acids. So this is an example of a primary protein. It's a straight chain of amino acids. So each one of these blocks represents an amino acid, and in between we have a peptide bond. Peptide bonds are what joins amino acid to make the chain. So the long straight chain is again the primary protein. Secondary proteins can be either found in an alpha helix, which kind of looks like a curlicue, or a beta pleated sheet, which looks a bit more like a zigzag or a fan. So again, that's an either or. It's a little bit more complex than the primary structure. So here is a secondary protein that looks like the curlicue. This is called an alpha helix, so it can look like this, or it could look like this, like a fan, zigzag, also known as a pleated sheet. So a little more complicated. Tertiary structure is when the protein coils and folds, producing a three-dimensional shape. So that's even more complicated yet. So here's a tertiary protein. You can see it has coils in it, and they're all folded up. So it makes it a little bit more globular, a little bit more large. That's a tertiary protein. And then a quaternary structure is a protein complex produced by interacting polypeptide chains or subunits. So first let me say that polypeptides greater than 100 amino acids are called proteins. So if you see the word polypeptide, we call it a protein when there are 100 or more amino acids grouped together. So these are bigger. But the quaternary structure is a protein complex produced by interacting polypeptide chains. So lots of polypeptide or multiple polypeptide subunits grouped together to make a very complex protein. So you can see that these individual polypeptide subunits, you can see some blue and some purple, are interacting to form a larger molecule. And an example of this protein would be hemoglobin. And hemoglobin is pretty critical. This is what carries oxygen in the blood. So proteins are huge. And when I say huge, I don't mean size-wise necessarily, but they're huge in importance. They're very important in the body. They make up a ton of what makes us up. Proteins are found in the skin, in the lens of the eye, in the muscle of the heart, the skeletal muscle, the smooth muscle. They make up your hair, your nails. We could go on and on. So proteins are critical in the human body. So if a protein gets pushed out of its optimal homeostatic range, and then it is either permanently or temporarily going to lose its shape, we call that denaturation. So again, it's a change in shape of the protein that can be temporary or permanent due to an extreme in heat or pH. And we're going to add a little bit to that actually. So denaturation or loss of protein shape can be caused by extremes in pH. Now this could mean too acidic or too basic. Temperature, which again could be too hot or too cold. UV light. And salt levels. If any of these are out of normal range, it can cause a protein to become permanently or temporarily denatured, which means that it will lose its shape. And if it doesn't have the shape that's so critical that we just went over, then it can actually be unable to function. And imagine in the human body, proteins like hemoglobin, how greatly that could affect its ability to carry oxygen if it were to lose its characteristic shape. Nucleic acids are our final organic molecule. They are large, they store and process information, and there are two types, deoxyribonucleic acid or DNA, which determines your inherited characteristics, helps to direct protein formation, controls enzyme production because enzymes are proteins typically, and will help control your metabolism. RNA or ribonucleic acid will control the intermediate steps in protein synthesis or protein formation, and we know now how important proteins are. So, structure of nucleic acids. DNA and RNA consist of long chains of nucleotides which contain a sugar, either deoxyribose or ribose, a phosphate group, and then nitrogenous bases like A, G, T, C, and U. So let's break that down a little bit. Okay, so we're going to organize this into two sections, DNA and RNA, so that we can kind of compare and contrast the two. Okay, so DNA is made up of a sugar phosphate backbone, and the sugar is deoxyribose. Now, if we were to describe what DNA looked like, we would describe it as a double helix, and it also is going to have in it nitrogenous bases. And the nitrogenous bases are the letters that we often see in diagrams of DNA. So that would be A for adenine, T for thymine, G for guanine, and C for cytosine. Okay, and remember that A will bind with T and C will bind with G in DNA. So then we've got RNA, which is also made of a sugar phosphate backbone. The sugar is ribose, and RNA is a straight chain rather than a double helix. It too has nitrogenous bases, or those letters, and they are mostly the same. We have adenine again. Instead of T, we have a U for uracil, G, guanine, and C again, cytosine. And A will bind with U, and C will still bind with G. Here is a picture of RNA versus DNA. RNA on the left, single strand. DNA is a double helix. Looks kind of like a ladder that's been twisted around. And finally, high energy compounds. Now this is very important throughout physiology because ATP is the energy currency of the cell. ATP at the bottom, adenosine triphosphate. It's a high energy compound that contains 3 phosphate groups, which is why triphosphate is part of its name, tri meaning 3. Adenosine diphosphate contains 2 phosphate groups, di, and adenosine monophosphate contains 1 phosphate group, mono meaning 1. This concludes Chapter 2, Review of Chemistry and Biochemistry. See you in Chapter 3.